Vesicular transport. Vesicular transport mechanisms are involved in the transport of macromolecules such as large protein molecules, which can neither pass through the membrane by diffusion nor by active transport mechanisms. The vesicular transport mechanism includes endocytosis, exocytosis, and transcytosis. The first one is endocytosis. Endocytosis is the process in which the substance is transported into the cell by infolding of the cell membrane around the substance and internalizing it. It is further categorized into three types. This is the diagram for endocytosis, wherein this is the endocytic vesicle, and here there is fusion of the non-cytoplasmic sides. The first one under endocytosis is pinocytosis, that is, cell drinking refers to the process of engulfing liquid substances by the enfolding of a cell membrane. For example, reabsorption by renal tubular epithelial cells. The second one is phagocytosis. Here, cell eating is the process of engulfing of solid particles such as bacteria, dead tissue, and foreign particles by the cells. The process of phagocytosis involves three steps. The first one is the attachment stage, the second one is the engulfment stage, and the third one is the killing or the degradation stage. So, under endocytosis, the first one was pinocytosis, the second one was phagocytosis, and the third one is receptor mediated endocytosis. In receptor mediated endocytosis, in this process, the substance to be transported binds with the special receptor protein which is present on the cell surface. The receptor protein substance complex is then engulfed by the cell membrane by the process of endocytosis. Transport of iron and cholesterol into the cells occur by this method. Under vesicular transport, after endocytosis, the next one is exocytosis. So this is the ICF, we have the ECF, the cell membrane, and the cytoplasmic sides of the two membranes fuse. Exocytosis is the reverse of endocytosis. That is, by this process, the substances are expelled from the cell without passing through the cell membrane. In this process, the substances which are to be extruded are collected in the form of granules or vesicles which move towards the cell membrane. Their membrane then fuses to the cell membrane. The area of fusion breaks down, releasing the contents to the exterior and leaving the cell membrane intact. Release of hormones and enzymes by secretory cells of the body occurs by exocytosis. The process of exocytosis requires calcium and energy along with docking proteins. The third one under vesicular transport is transcytosis. Vesicular transport within the cell is called transcytosis or cytopempsis. It is quite similar to endocytosis and exocytosis. Three basic steps involved in this process are vesicle formation, vesicle transportation, and docking in the cell. Now, apart from endocytosis, exocytosis, and transcytosis, there are other transport processes which occur across the cell membrane, that is, movement of substances between the intracellular fluid and the extracellular fluid, that is the ICF and the ECF, through the cell membrane. There are many situations in the body where transport of substances occurs through the epithelia and the capillary endothelial cell membrane. Some of these processes are: the first one is transport across epithelia, which involves movement of substances from one side of epithelium to the other. This type of transport occurs in the GI tract, the renal tubules, pulmonary airways, etc. Here, the cells need to be bound by tight junctions, and have different ion channels and transport protein in different parts of their membrane. And the second one is ultrafiltration. When a solution of protein and salt is separated from plain water or a less concentrated salt solution by a membrane permeable to salt and water and not to the protein, there will be a net movement of water on the protein side by diffusion and movement of salt away from the protein side. This process is called dialysis. Ultrafiltration refers to occurrence of dialysis under hydrostatic pressure. This process occurs at capillary level in the body. The capillary blood is under hydrostatic pressure. The pressure is 35 mmHg near the arteriolar end and gradually declines to 12 mmHg near the venous end of the capillary. Lastly, ultrafiltration plays an important role in the formation of body fluids.